So our lab is interested in epigenetics, specifically reproductive epigenetics, and trying to understand how the epigenetic regulation of the germline affects both fertility as well as embryogenesis and long-term health and development of offspring. So the lab looks right now specifically at what's going on in sperm and the paternal germline, more specifically how little proteins called histones and their post-translational modifications are altered during spermatogenesis, how that packages the chromatin and the DNA in that sperm cell, and then how that influences and regulates what's going to go on in the early embryo, and then how that affects development later on in life. So we work in several different mammalian species. We work in mice, cattle, and humans. We do a lot of different techniques. So we have a lot of genomics. We have genetic mice models. We also have a lot of cell biology, imaging, IVF, other types of molecular biology as well. So when students come in, I like to kind of assess where they're at. Do they have a fundamental understanding of what's going on with the research program? Sometimes I get students that maybe have a background in reproductive physiology, maybe genomics, maybe neither, and that's completely fine. And so I try to figure out where a student is at with regards to their knowledge base, with their skill set, and then kind of develop a plan from there. So if someone who is relatively new to this area, then it's a matter of trying to bring them up to speed on readings, the basic kind of just understanding of the field, both of chromatin biology and epigenetics, but then also reproductive biology. If it's a technique-based situation, then we need to make sure that they understand the techniques. And so oftentimes that first year or two is really just trying to get a good grasp of what we're doing in the lab. And then from there, really pushing students to kind of develop more of an independence where they're able to kind of plan their own experiments, maybe think about new techniques, new methods that they can kind of bring in, and have a little bit more ability to really drive that project by themselves. And then hopefully by the time that they're finished, they're really at that point where they've kind of mastered that area that they're working on, and they can go from there. I try to be really upfront with students when they start, what is it that you want to do? And fully acknowledging and understanding that that may change from the time that they start their PhD to the time that they graduate. That's normal, completely normal. And so I try to work with students throughout that kind of time frame to try to see, where are you at mentally? What are you thinking you want to do? What are your interests? And then trying to help and adapt to what they want to do. And all along, trying to make sure that they have a broad skill base that one is going to be usable regardless what direction they go. So things like being a good communicator, being a good scientist, technically solid, being able to write. Doesn't matter what direction you're going. I think those are skills that you have to develop. But then from there, if you need to pick up more specific skill sets. So if you want to go into academia, if you want to continue a postdoc, making sure that you're getting a lot of grant writing experience on top of that. Making sure that you are getting more mentoring experience. If it's something where you want to go into industry or policy, making sure that we're reaching out to people to put you into the right connections so that way you'll be able to get those jobs. And so really just trying to work with the student to see what they want to do so that way they're going to be best prepared to go whichever direction they end up going.